Hello everyone, welcome to my video series on GC bias and GC correction. Now GC bias is one of those topics which are not discussed in great detail in any video or in any tutorial that I've seen. And it is one of those concepts which are crucial for sequencing and for people working in bioinformatics. So this series is dedicated to clearing out those concepts so that we don't have to struggle with those ever again. So let's begin the part one of our video series where we talk about introduction of GC bias and GC correction. So without further ado, let's dive in. So first of all, let's look at this very small sequence consisting of the four bases A, T, C, and G. You can see that there are five G and C bases. So GC content here will be 5 divided by 10 because there are total 10 bases right here. And the GC content here is 50%. So this is what GC content is. There is no rocket science going on here. Now, let's go forward. Suppose this blue line right here, the straight line right here, is our genome. And we divide this genome into small equal-sized bins. So these are equal-sized regions. They can be 1 kilobase or 100 kilobase, 10 kilobase, etc. But they have to be equal. Now you can see that each of these regions have different GC content level. For example, this region right here has 70% GC content, which is pretty high. But this region right here is actually much lower in terms of GC content. Now above you can see the number of reads we get after sequencing from each region. For example, from this region we get 100 reads, but from this region we get only 60 reads. And from this region we get even lower number of reads, for example 35 right here. Now let's look at this even more carefully. You can see when the GC content is close to 50%, for example these green ones, you can see that the number of reads are relatively higher. But when the GC content level is much higher than the 50% or much lower than the 50%, then the number of read count actually reduces quite a lot. So that is something you should hang on to at first because that is the basis of GC bias. So first of all, when we do sequencing, when we do whole genome sequencing, the reads are sequenced uniformly from the whole genome. What does that mean? That means we are supposed to get around equal number of reads from equally sized genomic regions. That means each of these regions should approximately give us the same number of reads. But this is not happening here, as I showed here right now. And the amount of reads from a particular region actually can depend a lot on the GC content of that region. For example, you can clearly see right here. So the sequencing rate, the rate at which we are sequencing may be low for GC rich or AT rich regions. For example, you can see that it is low right here in the GC rich region, also low right here in the GC rich regions, and also here where these are GC poor. GC poor means AT rich. If there is only 31% GC content right here, then the AT content will be 69%, right? It will be AT rich. So in these cases, you can see there are lower number of reads obtained. So let's actually see what we can make from this nice little table right here. We can make a curve such as this. So GC content percentage will be in the x-axis and normalized read count will be in the y-axis. So what is normalized read count? So these are raw read counts. You can divide them by their sum or you can divide them by their mean. So you can normalize them in different way. But whatever you do, you will be able to get such a nice little bell curve. That means areas where there are plenty of GC content, we are getting lower number of read count. And also where we have low amount of GC content, we are also getting lower number of read count. Gradually the read count increases as the GC content goes towards the 50% mark. Okay, so this is GC bias. There is no rocket science going on here. And if you have understood so far, you have now understood the basic concept of GC bias. So now let's investigate why this GC bias actually occurs. So this is your entire genome, the DNA. Now during sequencing, what happens is this DNA is fragmented using shotgun fragmentation or shotgun sequencing. So we create small fragments, then some adapters are actually attached for sequencing at the both end of each fragment. The next step is the crucial one. Now, very often in most experiments, the amount of DNA that you see here is not enough for sequencing because the input DNA is actually very low often. So for better sequencing, for higher quality sequencing, what we need to do is we need to amplify, we need to copy these fragments multiple number of times to increase our input DNA amount. So that is where PCR amplification comes in. So PCR means polymerase chain reaction. So in PCR amplification, we make multiple copies of the same fragments and keep them in our sequencing solution. So PCR amplification is the major source for GC bias. So why is that? It is because PCR amplification does not work equally well for all fragments. So maybe for some fragments it will create 10 copies. For some fragments it can only create 5 copies. And that often depends on the GC content of those fragments. This is why the number of fragments increase or decrease artificially, not because of any biological issue, but because of the variation in GC content at the different genomic regions. So now you understand why GC bias occurs. So next, we are going to see how GC bias varies between sample to sample and between experiment to experiment. Suppose we have three healthy samples with very similar biological property. Maybe they are all Indian people or Sri Lankan people, or maybe they are all American white people, and they are all healthy people. We do sequencing on them. We construct all three of their GC bias curves. You will be able to see that in the same lab, if it is done at the same time in the same lab, then the GC bias curves will be very close. There will be some difference, obviously, from sample to sample, but it will be close. But if we take those same three people and again do the sequencing in a different lab or in a different environment, then you can see that the GC bias curves now actually look very different from these. So this is a very common phenomenon, and this is called batch effect. Batch effect refers to a phenomenon where some curves or some analysis is actually very much influenced by the difference in environment rather than the difference in the biological property. And GC bias is one of the core backbone basically of the batch effect, which actually makes machine learning modeling very difficult to generalize. So just to summarize, the nature of GC bias can be very different from experiment to experiment, and this is called batch effect, which we are showing right here. And there can be slight difference in GC bias for different samples sequenced within the same experiment, which we are seeing right here. There are some differences. Here is the most interesting part. Suppose we take this red sample right here and we sequence it in the morning, maybe in day 1, and in day 7, we again sequence that same sample in the afternoon using in the same lab. Now you will be expecting that the two curves from those two sequencing will be exactly the same because they are the same sample, they have been sequenced in the same lab. But no, there will still be some difference because of the time difference and the difference in environment such as temperature, humidity, etc. That is also an interesting fact to understand. Now, obviously, we want to correct for this GC bias. So what does GC correction actually mean? So this is what GC correction looks like. So GC content percentage in the x-axis and coverage in the y-axis, we saw that this is the GC bias curve. When we do the GC bias correction using some algorithm, the GC bias curve will look something like this. It will become much flatter than it actually is right here. So when we do this kind of GC correction, we are going to see some very positive impact. For example, you can see that here in the upper side, in the top side, these are original uncorrected coverage profile. So what is a coverage profile? For example, you can see in the x-axis right here, we have genomic beans. So these are genomic bean numbers, bean number 600, bean region number 800, and so on and so forth. So these are bean regions. And on the y-axis, we have the normalized coverage, right? So you can see that the coverage, the normalized coverage for the different beans are actually very, very different. They are very heterogeneous. Now, in healthy sample, the coverage profile is not supposed to be heterogeneous. It is supposed to be very uniform across the whole genome. When we do the GC correction, you can see that after correction, you can see that the coverage profile is now very, very uniform. Now you can see that I have written here GC fix corrected. GC fix is a GC correction tool that we developed in our lab. I was the first author in this. So you can check out that work. You can use that tool as well to correct for your GC bias. It is well documented. Okay, so this is the cancer sample. So in cancer sample, you can have regions where you have amplification and you can have regions where you have neutral. So no amplification basically. You can also have deletion, but we are not showing that here. We are only showing amplification and neutral region. So amplified regions and neutral regions. Now you can see that before GC correction, the regions are actually mixed. They are very much like abrupt changes and very noisy. But after GC correction, you can see that they are clearly separating out and they are very uniform within those areas where the amplification is happening and where neutral region is taking place. So that is the impact of GC correction. So that is all about my first video on this. So in the next videos, we are going to talk about some methods of GC correction. So lowest pin based GC correction is the most popular one because it is very easy to understand. I will also talk about single position GC correction, which not many people really talk about, but it is a very important concept to understand and is actually better than lowest pin based GC correction. So see you in the next videos. If you like the video, feel free to subscribe and share. And also you can donate to our channel to the link in the description. Thank you very much.